Hello, everyone. Welcome to our British Society for Gene and Cell Therapy, Patient and Public Engagement webinar in this sunny evening. I'm Sonam. I am a postdoc researcher at Institute of Child Health, and I'm also leading the Public and Patient Engagement Subcommittee at the BSGCT, and alongside our subcommittee members, Ozgi, Raj, and Juan, we run these PPE webinars every 3 months on a Wednesday afternoon, a Wednesday evening even. And the aim of our webinars is to kind of bring gene and cell therapy to the public, create a platform for awareness and discussion with the general public and patients. And this is something we run every 3 months. And we look forward to your engagement and questions in the chat. So before I introduce our speaker for today, I just want to run through some housekeeping items. So you can send us your questions through the talk using the live chat window in the YouTube. To use it, you do need to sign in with your YouTube account or a Google account and then join the channel. These messages are not private, so everyone who tunes in can read them. Please do include your name and affiliation in your question if you can. And I will ask the speaker the questions after the talk. So the sooner you ask the question, the more likely we'll have time to answer them. So without further ado, I want to introduce Dr. Ben Jevans, who is working here at the Institute of Child Health at UCL as well, and who I've had the pleasure of knowing for the past year. And thank you very much for agreeing to give a talk today again in this really sunny evening. So to introduce Ben, he's a postdoc researcher, as I mentioned before, and he's dedicated his research to understanding the processes involved in nerve damage and developing cell-based therapies. And his work has covered spinal cord injury and Parkinson's disease. And over the past 5 years, he's focused on Hirschsprung disease, a congenital condition where children are born missing nerves in the distal bowel. And he strongly believes that real progress in treating such conditions depends on bridging the gap between research and the lived experience of patients and their families. And today he's here to elaborate more on that. And the title of his talk is How to Develop a Stem Cell Therapy. So yeah, I'll pass it on to you, Ben.
Okay, thank you very much. Let me know if you can't hear me or if there are any problems. Yeah, thank you very much, Sonam. As Sonam said, my name is Ben. I'm going to be giving a little bit of an introduction to stem cell therapies, including what we mean by that, what we can achieve with a stem cell therapy, and I'm going to show you the work that we've been doing to develop a stem cell therapy for Hirschsprung disease as an example of that. You have around 35 trillion cells in your body. That's a number that's so big, it's hard to comprehend it in your head. But if we were to line up all of those cells end to end, they would reach to the moon at least once. The vast majority of those cells have become highly specialized to a particular task. So a bone cell, for example, has been specialized to become very rigid so that muscle and tendons can attach to it and pull. A nerve cell has become specialized to transmit packets of electrical information to communicate with other cells. As a rule, this specialization, which we call differentiation, is a one-way process. Once a cell has become differentiated and specialized, it's stuck as that cell. And they also can't divide to make any more of themselves. We do now think that there are some cells which are an exception to that rule, but in general, it applies to all cells in the body. Stem cells, on the other hand, retain the ability to divide, multiply, and produce more of themselves. And they can divide asymmetrically so that they stay as a stem cell, while their daughter cell can be directed to form any specialized cell in the body. We call the most powerful of these stem cells, the ones with the most potential, pluripotent stem cells, with pluri meaning many and potent meaning strength. And you can see immediately how we could use these for medicine. Life as we know it could not exist without stem cells. The most powerful stem cell, the one with the most potential, is formed at conception when sperm meets egg. The resulting cell, which we call a zygote, will give rise to all 35 trillion cells within the body, including all specialized cells. The zygote divides to form the multicellular morula, and then the blastocyst, and eventually a fully functioning adult human, which will one day be able to produce sperm and eggs of its own and continue the cycle. And you can see immediately that the power and potential of these stem cells is at once absolutely crucial for life, but also potentially problematic if left unchecked. As the zygote produces more and more cells, we need to know that the vast majority of those are going to stay as the cells they become. We don't want all cells in the body retaining the potential of these pluripotent stem cells. You don't want, for example, in the adult, a cell in the lung deciding that it would like to now be a bone cell. And you don't want the cells that are beating in your heart deciding that they're getting a bit tired of beating and they'd rather go back to their nice quiet life as a stem cell. And by and large, this problem is addressed by lineage restriction. In general, this is referring to that one-way process of differentiation. So as an example, this is the stem cell niche in the bone marrow, and it's packed full of these hematopoietic stem cells. This is a type of stem cell which has had its lineage potential restricted. So it can still produce a lot of different types of cells, but they're all blood related. It can't produce any cells related to, for example, the kidneys or to a fat cell. There are other stem cells for that. This stem cell's job is purely blood related. So this can divide to produce either a myeloid or a lymphoid progenitor cells. Again, these are a type of stem cell, but they're now even more restricted. So the myeloid progenitor cell can only produce all of these cells on the left-hand branch. It can't produce any of these right-hand cells. And as we move down and down through the tree, the cells become progressively more specialized until at the end, they're what we call post-mitotic, which means they can't divide anymore and they've become completely specialized to a given role and they're stuck as that cell. And we can take advantage of this natural process when we develop stem cell therapies. But why do we want to develop them? Well, stem cells can be used for almost any kind of therapeutic application you can think of. They could be used, for example, to replace stem cells lost after an injury. So what you're seeing here is a cartoon of a spinal cord which has taken a blunt force trauma, a contusion injury, to the center of the spinal cord. And for various reasons, when this happens, the cells at the center of the injury die away and can't be replaced. And that breaks the connections that these spinal cord cells were making. And when those circuits are broken, the result is usually paralysis. Stem cells could be used to repopulate this fluid-filled cyst and potentially restore those circuits so that we can enable function again. We could also use stem cells to treat neurodegenerative diseases. So what you're seeing here in this little icon is a slice through the brain about the level of your eyes and ears. And here you can see in this dark color, a special region of the brain called the substantia nigra. This is absolutely packed full of dopaminergic neurons, and they're called that because they produce a chemical called dopamine. And the job of dopamine is to lubricate our movements and to help you initiate movements. In Parkinson's disease, these dopaminergic neurons begin to die away. And that's where you get the symptoms of the characteristic rigidity and tremor occurring. If we were to transplant stem cells in, then hopefully they could replace those missing dopaminergic neurons, produce new dopamine, and help to treat the symptoms. We can also use stem cells for more unusual purposes. Stem cells can be engineered to express certain genes, molecules, even drugs, and then they could be transplanted back into the patient where they would continue pumping out the drug, in which case we could continue thinking of them like a little drug factory. Moving away from a direct application, we can also use stem cells to study disease in a dish. So if we take a patient's stem cells who has a genetic disease, all of the stem cells that we take and we expand in the dish will have that same genetic problem. And we can then study how that problem impacts the cells. We can test new therapies. And one day it may even be possible to treat those cells in the dish with a gene therapy to correct the problem and then transplant those back into the patient. And ultimately, all of this is useful for developmental biology. The more we understand about how stem cells work in health and disease, and we're able to treat them, the more likely it is that we'll be able to intervene in developmental processes that go wrong. So stem cells offer an incredibly therapeutic medical product, and we really want to be able to harness their potential. Any stem cell therapy that we develop would need to meet a few criteria. So firstly, we need to know that the stem cells would stay where we transplant them. Some types of stem cells are surprisingly migratory. They like to move around. And that's good to a point. We want the cells to move through the tissue and enough to fix the problem. But we don't want them leaving the initial tissue we've transplanted them into and going to another location where they might cause a problem. In a similar vein, we need to know that the cells that we transplant will only become the cells we want them to. So if we take this, a patient who's had a stroke, for example, and we transplant stem cells in, we need to know that they're going to become brain cells and not something like a bone cell, which would become very problematic. We also need the cells to expand, but only to the extent they need to. We want them to multiply enough to fix the problem, but then stop. We don't want them forming tumors. And finally, of course, we need them to actually fix the underlying problem. The process of turning anything into a medicine is long and complicated and therefore expensive. But this is necessary. There have been instances in the past where treatments have been used for a context that they weren't adequately tested for, and the consequences have sometimes been severe. Any therapy that we now try to develop starts in the research and development phase, in which we study what's going wrong and we identify a potential solution. We then proceed to preclinical studies where we will test that potential solution in vitro, meaning in a very, very controlled environment like a glass test tube or a glass dish, and in vivo, usually meaning in an animal model. And here we're looking at whether that potential solution does indeed treat what we're hoping it would treat. Assuming that goes well, we proceed to clinical trials. These are normally structured in several phases, with the first phase mostly looking at how well tolerated the new therapy is by a patient, and later phases evaluating how effective that treatment is, usually, if possible, comparing it to an existing treatment. And if that all goes well, then we proceed to review and approval, where it's essentially packaged into a medical product. All of these phases can take many years and cost a lot of money, with the cost of clinical trials typically reaching 10 to hundreds of millions of pounds. In our lab at the Great Ormond Street Institute of Child Health, we are currently at the preclinical studies phase of testing our stem cell therapy for Hirschsprung disease. And I'll spend a lot of the rest of the talk discussing the processes that we're going through to try and prove that this would be a viable therapy. Hirschsprung disease, to give you a bit of background, is a relatively rare condition affecting around 1 in every 5,000 live births. The main symptoms are severe constipation and a risk of infection. And these symptoms are so severe that most patients will require surgery within the first year of life, often when they're only a few months old. And it's thought that the vast majority of these symptoms arise due to an impact of the disease on the enteric nervous system. So we're all obviously familiar with the brain in our heads, but scientists now recognize that we have a second brain in our guts, which we call the enteric nervous system. Unlike the brain in our heads, which is squeezed into a solid lump of brain tissue, the brain in our gut is spread out like a fine net all the way through the intestines. If we were to take a cross-section through the intestine, you'd see this layered structure like an onion. The innermost layer is the mucosa. That's formed of these fine finger-like projections that help to absorb nutrients and water from the food and drink we consume. We then have 2 layers of muscle, and sandwiched between them, shown here in yellow, we have the enteric nervous system. If instead of cutting a cross-section of the intestines, I were to cut it open along its long axis, as this toilet roll tube has been opened here, and I were to then stain the enteric nervous system so that we could see it, it would look a little something like this. So what you can see here are nerve cells. Each one of these little pink dots is a single nerve cell or a neuron, and the light blue lines are the connections those nerve cells are throwing out to one another. This is a piece of intestine about 0.5 centimeter long, and you can see that even in this tiny, tiny amount of your intestines, there are hundreds and hundreds of these cells. And these millions of cells that are spread all the way through your intestines are absolutely vital for the intestines to do their job directed by the enteric nervous system. The enteric nervous system controls the release of digestive enzymes, and importantly, for the setting that I'm looking at, it tells the muscles of the intestines when to squeeze and when to relax. The enteric nervous system is derived entirely from a special type of stem cell called a neural crest cell. This is another example of a lineage-restricted stem cell. It doesn't form all cells like a pluripotent cell, just a select few types of cells. Early in development, around the 4th week of conception, these cells have arisen at about the level of the spinal cord, and they migrate through the body to reach the developing gastrointestinal tract, shown here in purple, at about the level of the mouth. And they then move all the way through our intestines, from the mouth all the way through to the bottom, and develop into the enteric nervous system. In Hirschsprung disease, the movement of these cells through the intestines is compromised, leaving a variable amount of the distal bowel aganglionic, meaning it's missing nerves. And this is what causes the symptoms of Hirschsprung disease. As I mentioned, the nerves tell the muscles of the intestine when to squeeze, when to relax. And if they don't receive that input, they just squeeze tight. And then the area above the squeezed region begins to swell with accumulated poo, and that can even rupture if it's left untreated. In our lab, we're focused on trying to repopulate the missing nerves with stem cell-derived nerves to try and restore function. If we're going to try and do this, then it makes sense to use the cells that nature uses, those neural crest cells. So to do that, we need to turn pluripotent stem cells, the ones that can produce anything, into neural crest cells. And we do this by following a protocol or a recipe, which over the course of 6 days, we add various supplements or ingredients. We have found that adding different supplements will encourage these pluripotent stem cells to form neural crest cells. Over many years of research, this was led by a group up in Sheffield, led by Anestis Tsakiridis and Peter Andrews, who studied how neural crest cells are formed in the body, and they then developed a protocol to mimic that in the lab. And they found that adding these specific supplements in specific quantities at specific times will activate all of the genes that are needed to transform pluripotent stem cells into neural crest cells. I'll just say here that all of the stem cells that I'll talk about for the rest of the talk are neural crest stem cells that we made by following this protocol. And I will also say that I've labeled them, or they have been labeled rather, I didn't do it, with a fluorescent green marker so that we can see where the cells are when we're testing them. Obviously, before the neural crest cells that we've made get anywhere near a patient, we need to check that we've made what we hope to make. And the way we do that is by looking at whether the cells are expressing the genes that they should. So I mentioned that the stem cells will start expressing genes as they differentiate to a given type, in this case, a neural crest cell. And one of the genes that we know neural crest should express is SOX10. So what you're seeing here is the results of a few experiments in which we tested 4 different stem cell lines, meaning, for example, that they came from a different donor, but they all started as pluripotent stem cells. And we have driven them to form neural crest. And here we're looking at how many of those cells actually express SOX10 by the end of the process. And you can see that no matter which stem cell source we started with, the vast majority of them do express SOX10. But that's not nearly specific enough if we're going to put these into a patient, because there are actually 4 different types of neural crest cells. I mentioned that these develop along the spinal cord. Of course, your spinal cord is quite long, and depending on the exact area that they come from, the neural crest cells will be slightly different, and they might be able to produce slightly different cells. So neural crest cells that arise at about the level of our head make cranial neural crest. Neural crest cells arising from the neck and shoulders make vagal neural crest. The chest and abdomen make trunk, and the hips make sacral neural crest. We're predominantly interested in vagal neural crest. These make enteric nervous system cells. The numbers that you're seeing on the right here refer to Hox genes. We don't need to know a lot about Hox genes for the purposes of this talk, except to know that they're involved in patterning the embryo, and the different populations of neural crest cells express specific signatures of Hox genes. So we can see, for example, that vagal neural crest should always express HOXB4, 5, and 7. And so when we perform an experiment to check that these 4 cell lines can produce these Hox genes, we see that all of them can after we have driven our cells to form neural crest cells. So this is very encouraging. So that quite nicely shows that we can make neural crest cells by following the protocol. But of course, when we transplant these cells into a patient, we don't want them to stay as neural crest. We want them to form the nerve cells and neurons of the enteric nervous system. Before we go near a patient, we can test that by continuing to culture cells in a dish and adding even more supplements, which we found will drive them to form neurons. To test after we perform this second phase of culture that we have made the right types of cells, we use a technique called immunohistochemistry. This technique relies on the antibodies that our bodies produce in response to an infection. Those antibodies that our bodies naturally produce are very, very specific. Each antibody will bind to a single protein that is found on the surface of a cell. There are some proteins that are only found on a single type of cell. For example, we know that nerve cells always have a protein on their cell surface called TUB3. So a TUB3 antibody won't be able to bind to any other type of cell because it doesn't have that protein. However, it will stick to all nerve cells like a magnet. And if we tag that antibody with a fluorescent molecule, we can make all the cells that express TUB3, all the nerve cells, glow. This is called immunohistochemistry. So here you can see that we've applied a pink antibody to the protein SOX10, that one that all neural crests should express. And here we've applied it to, we've applied a green antibody against TUB3, and you can see that it's made lots of neurons. So this is very encouraging. After we push the cells through that second phase of culture, they do make the nerve cells that we want them to make. Obviously, we don't just want them to look like nerves, we want them to function like nerves. And to do that, we've used a technique called calcium imaging. In this image, you can see groups of cells, and we have focused on the ones that are indicated by these little colored squares. In this graph, you can see the results of the experiment. And what we're doing is measuring the calcium within those cells. Basically, what we're looking for is whether they can respond to electricity. And the reason we do this is because nerve cells transmit information by transmitting little packets of electrical information. So what we're essentially doing here is giving a little electric shock, speaking their language, and seeing if any of the cells can reply. And when we applied that electrical stimulation, which is going to be indicated by a gray hash line, you can see that some of the cells immediately responded by releasing a lot of calcium. So they look like neurons, they express the right proteins, and they seem to be able to respond to electricity the way that neurons should. So this is very encouraging. All of the work up until this point was conducted on cells in a dish, but now we'd obviously like to test the stem cell therapy in a more relevant disease setting. I work at the UCL Great Ormond Street Institute of Child Health, and this is a laboratory which is next door to Great Ormond Street Hospital. Great Ormond Street Hospital conducts more Hirschsprung disease surgeries than almost anywhere else in the world. And during this surgery, the parts of the intestine that don't have the enteric nervous system, those aganglionic parts, are surgically removed, and the remaining pieces of the intestine that do have nerves are reattached together. We approach the patient's families to ask permission to use some of the tissue that's being discarded. And if we get that consent, then we take the piece of tissue and we peel it, like peeling an onion, to only leave the muscle layer, the layer that should have the enteric nervous system in it. We then cut this layer up into lots of little segments around 1 centimeter squared, and we transplant it with either our neural crest stem cell therapy or a sham therapy, which contains the liquid that the cells are sitting in, but no actual cells. And we then put these bits of tissue into a culture incubator and keep them there for 3 weeks to see what happens. I'll just say again at this point that all of these neural crest stem cells that we're using have been labeled with that ZS green fluorescent marker so we can find them again. So here you can see a picture I've taken using a fluorescent camera of a piece of transplanted tissue. The fluorescent camera can see the fluorescent label that the cells have really clearly, but it can't as easily see the actual piece of tissue. And so I've drawn around it with this white hash line. And in the middle, you can see the green glowing stem cells that I've transplanted. This image was taken 7 days after transplant. This was taken 14, and this was taken 21 days after transplant. And you can see that the cells are spreading further and further through the tissue with passing time. If I process these images using a special software and various algorithms, I can mark the spread of those cells even more clearly. And when I count that up over a lot of different transplant experiments, I can confirm that the cells do indeed spread more and more with time. And interestingly, if I look at a really high magnification, I can see that the stem cells seem to spread by following blood vessels within the tissue. So we previously showed when the cells were in a dish that they did have the potential to form those nerve cells of the enteric nervous system. I now want to check that they can do that again after we've transplanted them in. So firstly, I stained some of the transplanted tissue with the red TUB3 antibody. I then took an image of the exact same area of tissue, this time using a green fluorescent microscope, because the cells that I've transplanted in should be green. Fluorescent microscopy allows us to take images of the exact same area using different fluorescent colors, and I can then merge them together to see where the cells are in relation to one another. And so what you can see here is there are a lot of cells indicated by arrows that were both red, meaning they're nerves, and green, meaning they must have come from the stem cells that I put in. I also checked for another protein using a red antibody, a protein called S100 beta, and this is only found on glial cells. And glial cells are the other important cell of the enteric nervous system. They kind of support the nerves and make sure they're functioning well. And again, when I image that same piece of tissue with the green fluorescent color and merge it, I can see many cells that are both red and green. So the cells are forming the right type of cells of the enteric nervous system after we transplant them in. But we really want to know if they're improving the symptoms of the disease. And to do that, we turn to organ bath contractility. In this technique, the piece of tissue is suspended on very thin thread between a peg that doesn't move and a force transducer that can tell every time something pulls on it. And this whole system is then put inside a glass jar, which is filled with a solution called Krebs solution. It just contains a lot of salts and sugars that keep the cells happy. And we then bubble oxygen through. So what we're looking for is the ability of that tissue to squeeze and relax. Remember, the Hirschsprung disease tissue is missing nerves. So in theory, it just squeezes tight and can't relax. What you can see here is a typical trace from one of these experiments. So the sham transplant is in gray, and the green represents the stem cell transplanted tissue. Every time this line goes up, that's the piece of tissue squeezing. And when it goes down again, that's the piece of tissue relaxing. And what you can see is that the neural crest transplanted tissue can squeeze and relax to a much greater degree than the sham transplanted tissue. And when I go through and count up the area under all of these peaks and compare it across many, many different transplants, I can see that the stem cell transplanted piece of tissue is significantly stronger at that squeezing and relaxing than the sham transplant. If I then apply electrical stimulation, indicated by this little red bar, we can see that the transplanted tissue responds to a much greater degree than the sham transplant. So again, as when we were looking at the cells in the dish, this is sort of speaking the nerve's language and checking if they can respond. However, I can then take this a step further and apply it to tetrodotoxin. And when I do that, we see that the significant difference is lost. Tetrodotoxin comes from the ovaries of pufferfish, and it's a poison that stops all nerves from working. So the fact that we saw a greater squeezing and relaxing in the transplanted tissue, and that's then lost when we apply a poison that blocks nerves, suggests that that greater squeezing and relaxing was being driven by the nerves. So while we've got the cells in that organ bath, we can use pharmacology to try and tease out which specific nerve cell types we've made. Pharmacology is a fancy way of saying we apply different drugs to the tissue when it's in that organ bath and see what happens. And the reason we're doing that is that there are lots of different types of nerve cells, and we want to be sure that we're making both the ones that say squeeze and the ones that say relax. So this is, again, another one of these traces where we've electrically stimulated the tissue. And you can see that when I add atropine, that greater squeezing is much reduced. Atropine is a poison that very specifically blocks the type of neurons that tell muscles to squeeze. If I then apply L-name, that squeezing comes back a bit. L-name blocks a specific type of neurons that tell muscles to relax. And when, again, I compare these results over many, many transplant experiments, I can see that the stem cell transplanted tissue responds much more to these drugs than the sham transplant, which tells us that it has both more of the cells that say squeeze and the ones that say relax. So in summary, from all this work, we've shown that we can make neural crest cells using the protocols developed by our colleagues at the universities of Sheffield. We can transplant those stem cells into Hirschsprung disease tissue, and the stem cells survive and form the right types of cells, those neurons and glia that we want. And most importantly, they seem to fix at least partially the problem by improving the ability of the tissue to squeeze and relax. We would now like to move this work towards a medical product, but there are a lot of steps that we need to climb in order to make that happen. Firstly, we need to go through that protocol that we use to make the neural crest cells and make sure that it's GMP, or good manufacturing practices, approved. When we use that protocol, we use a lot of products that are animal or bacteria derived. If we're going to put this in a human one day, we need to replace all of those with human grade equivalents, and that's expensive and it will take a bit of time. We also need to show that we can make enough cells to treat a human. These were all conducted on very small pieces of tissue, and obviously we need a lot more cells if we're going to treat an entire human. And we need to check whether we could achieve a better result if we used more cells, or if we could achieve the same result if we use less cells. And finally, we need to determine if there are any off-target effects, if the cells go anywhere we don't want, or if they have any toxicities. To do this next body of work, we applied for funding to the UK Research and Innovation Hub. This is a government-funded organisation that distributes funding to promising research, and they have a particular funding opportunity that we applied to, which is called the Developmental Pathway Funding Scheme. And the idea of this is that it takes promising research and moves it closer towards clinical trials. This was the title of the bid that we put in: Development of a Human Pluripotent Stem Cell-Based Therapy Against Hirschsprung Disease. And a lot of scientists worked very hard on this bid, which went out for review by independent experts in the field. I'm very pleased to announce that this was successful, and we will be beginning this next phase of the work in 2027. As we take these next steps, it is vital to engage with the community to make sure that we are addressing the areas of disease that matter most to them. To begin this, we established Hirschsprung Connect, which is our focus group of families with lived experience of Hirschsprung disease. There are 4 families in the group, with 3 individuals in each family, including 7 individuals with a diagnosis of Hirschsprung disease. And in 2 of those instances, both a parent and child were affected. We met 5 times over the course of a year, and then after each meeting, I sent out a survey based on what the focus group had mentioned to make sure I captured everyone's views correctly. And I just want to quickly go through the 4th meeting that we had, in which we discussed how we would select the subgroup that could be used for first-in-child clinical trials. So Hirschsprung disease is a very variable disease, and we suggested 3 subgroups that we could use to the focus group. In some patients, the entire colon is affected, meaning it's missing nerves. In some patients, both the colon and the small intestine are affected. And in another group of patients, unfortunately, for reasons we don't fully understand, the surgeries don't completely fix all of the symptoms, and the patients will continue having ongoing symptoms. We're proposing that one of these 3 subgroups might be the optimal first group to receive a first-in-child clinical therapy. This is not an easy decision to make. As researchers, we would want the first stem cell therapy that we test to be in a group that has the best chance of success, because that will give us the best chance to attract funding so that we can push our stem cell therapy through the clinical trials. But of course, that might not be the same group that has the most serious symptoms and the most desperate need for the therapy. We asked our focus group which of those groups they felt most comfortable with. About 50% of the focus group had mixed feelings about long-segment Hirschsprung disease, where the entire colon is affected. Only 25% had mixed feelings about total intestinal Hirschsprung disease, where the colon and intestine are affected. But the entire group felt positive about the group in which the surgery had failed to treat the symptoms. We are still many years away from conducting these first-in-child clinical trials, but getting the views of the community at this early stage means that when we get to that point, we're ready to take their views on board and make sure they're incorporated moving forward. We're very keen to continue working with the community, and to this end, earlier this month, we held Hirschsprung Disease Awareness Day at UCL. We invited around 80 members of the Hirschsprung community to hear talks from clinicians, surgeons, researchers, dietitians, a psychologist, as well as other members of the Hirschsprung community. And we also hosted several stalls of relevance to the Hirschsprung community to sort of signpost support that's already available. This was a really positive day, and after it was finished, I sent out a survey to try and collect some feedback on how the community thought it had gone. Around 14 of the 28 families have so far given feedback, and you can see that the vast majority of them found the Hirschsprung Disease Awareness Day very useful, and most of them learned a lot, with a couple learning a little. And importantly for us, after attending the Hirschsprung Disease Awareness Day, 86% of the families who've responded felt more informed about research. Of particular importance to me, I asked, before and after attending the Hirschsprung Disease Awareness Day, how positively did you feel about stem cell therapy? Before, 36% felt very positively, but after attending, 79% felt very positively about stem cell therapy for Hirschsprung disease. And that shows the difference that we can make with engaging with the community. And we hope to continue engaging with the community so that they can guide the research into the areas that matter most to them. So I'll just stop here and acknowledge the many people I work with, both at the UCL Great Ormond Street Hospital and our collaborators at the universities of Sheffield, Padua, KU Leuven, and Alderhay. I'd like to give a special thanks to the GOSH surgical teams, GOSH histopathology, the Hirschsprung patients and their families, and the Hirschsprung Connect focus group. And of course, thank you to our funders for allowing us to do this research, and thank you very much for listening.
Thank you very much, Ben. That was very interesting, and yeah, always enjoy your talk. So yeah, we can start with a couple of questions. Actually, the first question I had was sources of these cells. So the ones that you currently been using for your research, where do these cells come from? And like, if you were then to move to patients, would you expect to use patients' own cells and then, or like, would you have a different source of these cells?
So this is a really important question. The source of stem cells will make a huge impact on how the stem cell therapy moves forward. So for example, there's three main sources of stem cells, as far as I'm aware. We could use autologous stem cells, which means that we would take stem cells from a patient, expand them in the dish, the culturing as I showed earlier, and then we transplant those same stem cells back in. That's autologous therapy. And the advantage of that is that your body will recognize that the stem cells you're putting back in came from it. So your body obviously responds very strongly to anything that isn't from the self. So if you get an infection, your body will fight to kill it. And that can happen with stem cell therapies, but autologous therapies circumvent that problem because all of the stem cells came from you. The other option is allogenic therapy, where the stem cells might come from a donor. So for example, if I had a problem, a genetic problem that meant that I couldn't use my own stem cells, but you didn't have that problem, you could provide a donor source of stem cells. That would circumvent my genetic problem, but now my body is much more likely to attack the stem cells you've put in because they didn't come from me. There are a couple of ways we can get around that. So for example, either I could be on immunosuppressant drugs for the rest of my life, which would mean that my body wouldn't be as able to fight off the stem cells, but then I'm much more vulnerable to infections. The other way is that we can try to match, I think they're called human leukocyte antigens. It's HLA matching. And this is basically, it looks at which proteins are on the surface of your cell. So when our immune system is trying to work out if cells are self and therefore should be left alone, or if they're not self and should therefore be destroyed, it reads these HLA proteins that are on the surface of a cell. There are only a certain number of those. They're not infinite. It's not one of those situations like a fingerprint or an iris where they're individual to every human. So we could, for example, make sure we match the HLA antigens to try and reduce the risk of our body fighting it off. And the final source is induced pluripotent stem cells, where the cells have come from a donor and but they weren't stem cells to begin with. We've sort of taken a skin cell, for example, and we've then transformed that into a stem cell. And that's the cells that we are currently working with. So we've got a couple of specific lines that we work with. But as we move forward into to getting this closer to clinical trials, we'll need to use specific stem cell lines that have already been approved and found to be safe. For example, they don't have any genetic instability. So it's an excellent question, and it's an ongoing work to find the best stem cell source.
Actually, on that note, actually there was a question. Depending on your, I guess it kind of follows on from that question of whether where the cells would come from. Would you expect there would be differences in the interaction of these cells with the rest of the enteric nervous system when you kind of transplant them in, or would the cell type affect that, or generally would you expect them to act as an independent half, or like integrate into the existing system within the patient?
That's a very good question. I mean, the honest answer there is I don't know. By and large, what we would hope for is that our protocols would be so robust that when we drive a stem cell to form, for example, a neural crest cell, we would form the exact same neural crest cell every single time. But that's not what we see in reality. We do see subtle variations, which we are looking closer at to see if we can sort of annihilate that variability. And if we're going to make this into a medicine, then we need to be very on top of that, and we need to define what variability is allowed. If we take cells from a different patients, would they respond differently? Honestly, I have no idea. What we have found in terms of interacting with the enteric nervous system is that when we transplant cells in, they seem to sort of home in to the enteric nervous system. So if there's anything left in Hirschsprung disease, which often happens in what we call the transition zone. So because the neural crest cells make it most of the way through, usually, that part of the intestine will be ganglionic. It will have the nerves. Then there'll be a slight transition area where it doesn't have as many nerves. And then there'll be the aganglionic area with none. Even if we transplant it into a ganglionic area, the cells seem to integrate quite happily into the existing ganglia, these little clusters of cells that the enteric nervous system is known for. And honestly, we don't understand why that happens. You would think that the clusters have enough cells, they have all the cells they want, they don't need any more, thank you. But they do seem to permit, if that's the right word, the stem cells coming in. And that again is something which we're hoping to look at in more detail in terms of looking at different cell types. I've never thought of that, and that is an excellent question that I will definitely be looking at moving forward.
Cool. There's a question in the chat. Is there any difference in longitudinal versus circular contraction in the effects of the disease or the therapeutic effect?
Oh my goodness, that is an excellent question.
Yeah.
That I definitely am not well placed to answer. So there are a lot of researchers doing excellent work on how contractions propagate, how they continue along the colon, and exactly which elements contribute to that. So there are excellent scientists. Grant Hennig is doing fantastic work on that, as is Nick Spencer, that I'm aware of, as well as many, many other scientists. In terms of which is more affected in Hirschsprung disease, I'm honestly not sure. I would imagine it's both, because in the aganglionic region, you don't have any nerves at all, which means both the circular and longitudinal muscles will be affected by the loss of enteric nervous signaling. In the transition zone, where you have a bit more patchy networks, it might be that there are some areas where the circular muscle is affected and some where the longitudinal muscles are affected. But that is really not my area of expertise, I'm afraid. So I'm sure that's a bit of a disappointing answer.
No, I'm sure they're happy with the answer. I had a question actually going back to one of the slides where you kind of listed the sort of things that you want to address moving forward. The safety aspect, like I guess when you were giving the talk, I was thinking, is there still a risk that these cells can migrate beyond the region of interest per se? And is there still a risk that these cells could become something else as well, rather than just sort of the cells specific to the enteric nervous system? And how do you plan to kind of study that? Like, yeah, because at the moment, as you said, you're working on cells on a dish and you had some patient samples as well. But yeah, how do you kind of foresee moving forward to study the safety aspect?
Okay, so I may forget parts of that
Yeah,
question. So
that's
if I
okay.
do, please remind me. I'll say firstly that there is an excellent scientist we worked with in the past called Julie Cooper, and she published a paper from our lab which addressed a lot of these problems. So she used an animal model and transplanted in neural crest stem cells. This was published, I think, back in 2016 or so. And in that, she looked at long-term safety and engraftment. So I think they waited for about 2 years after transplant. And what they were looking for was whether the cells migrated anywhere they're not supposed to, and whether they formed any tumors, because neural crest cells in particular are very migratory. I mean, they arise at the spinal cord and move all the way through the body. So we're interested in the neural crest cells that migrate to the intestines. Other neural crest cells contribute to the structures in the head, just about every tissue in the body. So it is a concern. In that paper, if I'm remembering rightly, they found no evidence of migration or tumor formation, which is the other concern, that the stem cells will divide more than you want. So that's a good indication. We'd need to test that more thoroughly, especially as if I remember rightly, I think that was an allogenic neural crest cell. I think they took neural crest cells from the intestines of one mouse and put them into the intestines of another. So ours is a slightly different stem cell therapy. We're using induced pluripotent stem cells. So we might need to repeat that test. I don't believe in that paper, and Julie will be very angry with me if they did, and I have now misremembered it. I don't think they checked for the potential to form other cells. And that is another important consideration, because although neural crest form the enteric nervous system, so the nerves, the glia that we would expect, neural crests in general are capable of producing many other types of stem cell. So we've hopefully restricted them down to the vagal neural crest, but we would need to perform extra tests to make sure that that's all they can make. There are various ways we can do that. So one of them, for example, is we could use fluorescent activated cell sorting, which is a technique in which we could screen the cells and say only the cells that express this protein on their surface, which we can confirm a neural crest, will be put into the medical product, and the cells that don't will be shoved to one side. So if you remember in that test early on, when Faye Cooper was looking at how many of the cells express SOX10, there was always a small percentage of cells that don't. We're not 100% sure at this stage what those cells are. So that fluorescent activated cell sorting would be a way of getting rid of that population so that we can be more sure of what they form. As far as I remember, the last time I checked, fluorescent activated cell sorting would potentially throw up more problems, because then you're exposing the cells to a more likely contamination as they move through I can't remember if it's still done with an air pocket or if it's now a liquid interface when they divide the cells. So we'd need some way of ensuring that the sterility of the cells was maintained, because obviously if you put those cells in a patient, they need to be very sterile. So the short answer is that's something we'll be looking at in the next phase of the work, potentially using animal models and longer periods of transplant. This was only 3 weeks. We'd look at maybe several months, and we'd maybe look at some sort of gating mechanism, like a threshold that the cells would have to reach in order for us to say, yes, we think these are safe and can only form the right types of cells.
Yeah. Okay, no, that's cool. I had a couple more questions for the end. These are going to be administered via surgery on patients.
So that the route of administration is something that is really important. And when you're trying to get a medical product through the various levels of regulation, the route of administration is something they look at very, very keenly. For a lot of therapies, you can just inject the stem cells into the blood, what they call an intravenous therapy. For things like liver stem cell therapies, that works really well. The stem cells go through the blood and they home into the liver very nicely. If we did that into the intestines, I'm not sure how well they would be able to home.
Mhm.
I will say that researchers in Paolo de Coppi's lab, Paolo de Coppi being a professor that we collaborate with a lot, are looking exactly at this question, and they're coming up with methods of administrating it. But I think that is quite early in the development, so I won't go too far into that, except to say that it is something that we're aware of and some very clever people are looking at it.
And then I think I'll kind of end with the final question. Because you're working on Hirschsprung disease, are you looking into kind of working into other diseases as well with these therapy? Well, open question.
So I guess it's maybe a tricky one to answer, because once we have a stem cell therapy, yeah, the temptation would be like, great, it works in this setting, let's use it for others. There's a disease, for example, called esophageal achalasia, which a very talented PhD student in our lab, Nanda Nesrakala, is working on. And in that, your esophagus is missing a very specific type of nerve cell. Specifically, it's the one that tells your muscles to relax. So that could be a very prime candidate for a stem cell. Maybe we'd need to gate it so that we're now only producing the relaxing nerves. But in reality, that first phase of the pipeline that I mentioned in an earlier slide, where you're looking at I can't remember what it's called now. The very phase where you're looking at the problem and then coming up with a solution. The solution tends to be quite specific to the problem. So in Hirschsprung disease, the reason we're able to propose a stem cell therapy is because a lot of excellent researchers did fantastic work in the past, and they have done an excellent job of characterizing what goes wrong in Hirschsprung disease. So researchers like Vasilis Paknis, Peter Vandenberg, Alan Burns, Nikhil Thapar have done amazing work. We now know what's going wrong in the tissue to a large extent. We don't have a full picture, but we know enough that we can say a stem cell therapy using this particular type of cells would probably treat the disease. So for any other intestinal diseases, we may have to go back to the drawing board a bit, decide exactly what's going wrong in that condition, and then whether this stem cell therapy would address that, or whether maybe we'd need to tweak it slightly.
Okay. Actually, on that, I had a question just popped up saying, do you consider target identification? I guess that's kind of going into similar kind of area, that if you were to go back into different, if you would have to go back into drawing board to find a disease that's kind of suitable, and maybe which something that, I guess this is where you always say it's good to kind of develop therapies, but we also want to understand the disease itself as well, which would help us understand how the therapies would potentially work or not in a particular disease. Yeah, I'm not sure. But
Yeah, absolutely. I mean, I guess on that, like, I've covered, or I've given an introduction to stem cell therapies as a cell replacement, but stem cell therapies could also be used, for example, to reduce inflammation. So there are some diseases where you get a lot of swelling and activation of the immune cells, which becomes problematic. You could also use stem cells for that, or you could use stem cells to secrete a certain protein that the body's missing. So it doesn't always have to be stem cell therapies, which I think might be what that question was about in terms of target and making sure you address it. And yeah, for each of those, you'd need to really tailor the stem cell therapy. So we're going specifically for a cell replacement therapy, but it's definitely not the only type of stem cell therapy that you could use.
Thank you, Ben. On that note, coming into 7:30 as well, I will kind of end the session here. And I just want to thank Ben for taking time to present your research and taking time to kind of explain. It's always difficult to explain, and hearing you talk is always quite inspiring. So thank you again for agreeing to give a talk, and thank you to the audience for staying with us till 7:30. And we'll have our next session in October, so we will kind of make sure to advertise that. And in the meantime, have a nice summer. and see you all in October, I guess. Thank you.
Thank you very much for having me.